what you're going to see is this, and now the skull is obviously gone. Okay, there is no bony part to this at all. The skull is missing. But you'll see this white layer right here. And I'm just going to pull on it just a little bit, not much. But right here, see this white layer? Now, that white layer is the outermost meninge, which means that it is the durameter. Okay, so that's the durameter. Now, it's been cut just so that's the only part left. In reality, this would encase the whole brain. Okay, we've actually got specimens years ago. We get the whole specimen and it'd have it all the way around the brain. So it doesn't just exist here; it's all the way around the brain. Okay, but that's all the part you're seeing. So you can cut off with scissors or something like that. So that's the durameter. Okay. Now, if you flip it right side up, okay, like this. All right. You can see very clearly. Two cerebral hemispheres, right and left, okay? If you run your finger over the surface of the brain, do you notice how you can feel risen areas and small depressions, right? Just run your finger over the surface of it, okay? There's risen areas and depressions, right? Feel that? The depressions, those shallow depressions, those are a sulcus or sulci, okay? The risen area above right here, this is a gyrus. Gyrus, sulcus. Gyrus, sulcus. All right? Now, if you take and do this with me, if you would, okay? Do you notice how you can kind of see there's a thin membrane right on the surface of the brain there? If you take your probe like this, take and poke, just poke it, okay? And then kind of pull up underneath it. Do you notice how there's a thin membrane there? See that? It's thinner than saran wrap, right? There, I had it. It tears very easily. Yeah, I'm tearing it. Okay. Come here.
Would that be the PIA?
That is the PIA meter right there. Yep, exactly, Becky. Okay, so that's the PIA meter right on the surface of the brain. Now, do you see all these black, brownish black structures running around it like that? See that? Now that shouldn't get in like that. It should continue on. But does anybody know what those are?
Blood vessels?
Those are blood vessels. Okay? Brain is very vascular, so those are blood vessels. They're on the list. It's just an extra. I'm just throwing that in because it shows that quite well. But the PIA meter is right attached to the surface of the brain right there. See how it's so thin? It's clear. It's a thin, clear layer. All right? Now, the arachnoid layer, for the most part, dissolves. When we put it in preservative, it tends to dissolve away. You know, if you were to take a spider web and put it in a bucket of water, you kind of shake the bucket, it would kind of dissolve in the water a little bit. But there is some of it left, and I'll show you that. Okay? And now let me do this. This is a cerebral hemisphere here, another cerebral hemisphere here. Down the middle, you can kind of see the start back here of the longitudinal fissure, right? Now, if you take and do this, take your thumb, works best, there's nothing sharp here, so nothing's going to hurt you. You take your thumb and start running it from, here's the cerebellum back here, start running it from here up. Now, do you see this web-like stuff in there? See, it's kind of webby in the middle? That's what's left of the arachnoid membrane, the arachnoid mater. Run this thumb about a ½ inch up. See, I'm going straight up the middle. See how it's going to tear, just like that? Okay, run it straight up the middle towards the front. See how it just separates? Just like this. Just run it right up the middle. Okay. Okay.
Uh-huh.
That felt good.
See how that separates? Now that, folks, see how that's much deeper? That is the longitudinal fissure. Okay? It's the longitudinal fissure. It's much deeper. Okay? Okay, so that splits the right cerebral hemisphere from the left cerebral hemisphere. Okay, and if we go back towards the back, now this is the cerebellum here. This deep depression right here, that's the transverse fissure. Okay, so you've got a transverse fissure here and a longitudinal fissure here. Longitudinal fissure, transverse fissure. Okay, longitudinal, transverse. Okay, this is the cerebellum here. All right? Now, let me do this. Okay. Now, let me do this with the cerebrum, right? And
You have to make sure
Kimberly.
you give me your email.
Let me ask you this. This is the cerebellum here. That would mean right here where I have the probe, that would be which lobe?
Occipital.
Exactly, that's the occipital lobe. And Chad, that would mean this one right here. Oops, let's go back here. This right here would be what lobe? Parietal lobe, right? Nick, this lobe right here would be temporal, and Nancy, that would be frontal. Piece of cake. Got it. Okay? It's that easy. All right? You guys know your skull bones, not a problem. Okay? Now, this back here is the cerebellum. Okay? Now, let me do this. Okay, take the cerebellum right here. Okay? And pull that down like this. Pull it down like that. Oops, that kind of tore a little bit, but pull it down like that. Like that, right?
Good job, you didn't do
Now, Kylie, you're from Austin, Minnesota, right? Oh, Claremont. Loverex, Austin name too. Well, you're from Claremont. What does this look like right here, Kylie? Right here.
I don't know.
Why are you giggling?
I have no idea what it looks like.
Okay.
What you think? I don't know.
All right. Chad, where are you from? Rochester. What does that look like, Chad?
If I was looking at the brain, I'd say it looked like our testicles.
Oh. Okay. You must have a pickup truck. All right. Okay. All right. Wasn't thinking that way, Chad, but in certain parts of the world, people go around and they expose their buttocks on Friday and Saturday nights, right? Just for funs. All right. To me, that looks like I'm being mooned. All right. Okay. To me, that looks like a buttocks. All right. And with that, again, sometimes associating it with things that you're familiar with makes more sense than just a couple of mounds. But
Yep.
what you have right here, there's 2 mounds here. Those are the superior colliculi right here. Superior colliculi. This one here and this one here below, those are the inferior colliculi. Now, these 4 mounds right here, those make up what's known as the corpora quadrigemina. A quad is a quantity of 4 in science. And these upper 2 are the superior colliculi, bottom 2 are inferior colliculi. Superior colliculi, inferior colliculi. That makes up a part of the brain called the corpora quadrigemina, which is part of your midbrain. Okay? We'll talk about the significance of what those do later on in the lecture, but those are the superior colliculi, inferior colliculi below. Okay? All right? Now, let's do this. And I've got to zoom this in a little bit, so give me just a moment here, okay? And open this up a little bit more, and you see up above the superior colliculi, you see this little glandular tissue right, it's hard for me to do this without getting myself in the way here. Right in the middle, right above the superior colliculi, see at the tip of the pointer right there? It's about the size of a kernel of corn, right there, deep in. That structure deep in above the superior colliculi, that's the pineal body or the pineal gland. That
think we
secretes
have
a
a better
substance
view
called
in ours.
melatonin. It's a hormone that's involved in wake and sleep cycles. Okay? So that's the pineal gland up there. So again, the superior colliculi, inferior colliculi, and then the pineal gland up above in between right there. Okay? Yeah.
If you're just referring to one, is it the superior colliculus then?
Correct. Yep.
Okay.
Collicula. Anytime you do the colliculi, that ends in I, it's plural.
Okay.
Like gyri or sulci. Yep, exactly. Okay. Good
Oh,
question.
oops.
All right, then you've got the cerebellum here, and then back here, this last part right here,
Spinal cord.
that's the spinal cord. Now, I have trouble magnifying this correctly because this camera doesn't do the right thing. As you look at yours, if you look down the middle of the spinal cord, there should be like a little opening down the middle of it. The opening down the middle, I know this is boring, I apologize, I just can't get the camera to focus on it. The opening down the middle is the central canal. Then if you notice in the middle, it's kind of tan. The middle part is tan. The outer part's white, right? Tan in the middle, white on the outside, all right? Okay. It's where it's myelinated and unmyelinated. Now, if you take the brain and flip it back over like this, okay? Now, there is something that sheep do better than we do, okay? And that something is they have a better sense of smell. I'm not saying they smell better than you do. I'm saying their sense of smell is better. And what you'll see right up here on the front, anterior part, right here, those are the olfactory bulbs. Now, I want you to think about this. You've all seen sheep before, right? The foremost part of a sheep's body is its nose, right? It sticks out there in the front, just like a dog, okay? And this large part right here is an olfactory bulb. There's another one over here. They're kind of oval shaped. Those are olfactory bulbs. They're able to smell, coyotes, dogs, wolves, whatever's trying to get them. Whether they can get away or not is another deal, but they can smell them usually in advance of the attack, all right? Depending on which way the wind's blowing. But olfactory bulb. And then coming off of that, there is a, mine doesn't show it as well. There it is. This is fairly big here on this one. See, this is the bulb right here, this oval structure right here towards the front. And then coming back from that, right here, this is the olfactory tract. The bulb, the tract, okay? Now, let me do this. Angelina, let me ask you this. Do you know, how do you think yours compare in size to this?
Probably smaller.
Yeah, it's much smaller. It's like half the size. Actually, it's about a quarter the size of a human being, all right? If you take a Doberman Pinscher, not a Doberman, a German Shepherd, or the drug-sniffing dogs that police use, they're about twice this size. Okay, you would never ever see a police officer going through a high school, putting their nose up to a locker, and right. I mean, you don't do that because they just don't have a good sense of smell. People don't, right? You send a dog in there, it's a different deal. Okay, dogs have a fantastic sense of smell and can track down drugs, criminals, or whatever. Okay, but olfactory bulbs, olfactory tract. Okay, now at these areas right here where it's kind of depressed right here, Ashley, do you know what used to reside right here where my fingers are?
Man, I can't think of it.
I'm sorry?
I said I can't think of it.
Okay, do you know?
I'm going to say the eyes.
Yeah, that's where the eyes were. Okay, that's where the eyes were at one point. Now, mine doesn't show this as well, but you see how there's kind of a V type, like a structure that comes across here like this? Okay. What's left here is the optic nerve. Optic nerve on one side, you got two eyeballs, right and left. All these are cranial nerves and they're paired. You have an optic nerve here, optic nerve here. This would have come out further and attached to an eyeball. Okay, eyeballs. But optic nerve, they mesh right here in the middle in an area called the optic chiasma. Now my brain's not that good at that point. But what happens is this. Let me make a quick diagram on the board here like this.
Do you guys have good optic nerves on yours?
Eye, optic nerve.
Yours is like ours then.
In the middle here, this X area, that's the optic chiasma. And then coming back here, right here and here, this part, that's the optic tract. Okay? Now, why this is significant, everybody, is this. When you see something as I look at you, you look at the board, whatever. When the neurons come from this side, some of them cross over, or some of them stay on the same side, some of them will cross over to the other side, okay? And so when people have a stroke and it affects the occipital lobe of their brain, that can affect their peripheral vision and depth perception. And so a lot of times, you'll see this with stroke patients, oftentimes when they go to get to a step, because on a step you have to do a step up, step down, figure out how much you have to bring the leg up or down to get to the next step, they'll struggle with that. And obviously if you can't do steps well, that can really affect your health, because you can fall down the flight of stairs and kill yourself, damage yourself badly. So sometimes they'll have problems with that. But what happens here is this, okay? Now you'll see this on this. Nerve, middle's the tract. We'll see, excuse me, the middle's the chiasma. We'll see the tracts here in a minute, okay? Then, as we go further with this, you see this kind of light tannish structure right in the middle right here, right there. That structure right there, that's the pituitary gland, okay? Pituitary gland. Now, it's not actually part of the brain, but it's attached to it, so we include it. And your pituitary gland secretes growth hormone, it secretes hormones that are involved with sexual reproduction, and it's a very important gland, okay? Pineal, or the pituitary. On either side of it, right here, and right here, that's the trigeminal nerve. Those will come off in just a minute, but those are the trigeminal nerves. Those have branches that go to your mouth, like when you get your teeth grilled. We're going to try and anesthetize branches of that so you don't feel the drilling going on, okay, if you have a cavity, all right? And then as we come back, we'll end up dealing with the medulla oblongata back here. Now, what I want you to do next, everybody, is this, okay? We're going to take and remove most of this duramater. All right, now in doing so, there's nerves underneath there that are running up towards the duramater. So what I want you to do is take your forceps in one hand, one of you, take your scissor in the other, and as you pull up on it, anything that's attached on the underside of the duramater, just clip it near the duramater, okay? Just clip it near the duramater, because there's some nerves that we want to see as we go up here. So be gentle. And the pituitary gland will come off.
I think those were optic nerves that we were looking for before.
There we go.
So snip those real close to the duramater. Yep.
Anything
And
that
that's
connects
our
on the underside of the
pituitary.
duramater, just clip it off to where it attaches. Okay, comes off like that. All right. Okay. Now, take that duramater, and remember I mentioned dura means durable, tough. Take it and grip it on the white. Take it and grip it and try and rip it. Okay, give it a shot. Okay, grip it, try and rip it. Just give it a shot. I've only had one person be able to tear this in 26 years doing this. It doesn't tear very easily at all. Okay, unless you get a fingernail in it or something where it's already torn. Take it apart that's not torn and try and rip it. It's tough. Okay, very tough membrane. Okay. Now, in pulling that off, we did pull off the pituitary gland and most of the trigeminal nerve. Now, let's go back to the brain here, okay, like this, okay, upside down, and let's go over those cranial nerves. Now, the cranial nerves are nerves that come off the brain and go to different areas and have different functions. We'll talk about those functions in the lecture later on. But olfactory bulb, olfactory tract, Now, the optic nerves came completely off when I took it off. What I have left right here, and I'll magnify it, what I have left right here in the middle is the optic chiasma, and see this part coming back like that? That's the optic tract, okay? Optic chiasma, optic tract. Where the nerves were coming out here, the nerves would have been sticking out like where the probe's at. Those came off when I cut the dermator off. Then you see these two flat ones here. This one here, and this one here, right? See those two? Those two are cranial nerves also. Those are called the oculomotor nerves, which, Anna Peters, that would probably mean they control what? I'm sorry? Of what? Oculo. Oculo is eye. Ocular. Okay, binocular. All right, so those control movement of your eye, the eye muscles. You know, you have to have coordinated eye movement to see single vision, right? Because if you see double, there's a problem. Okay, so those are the oculomotor. Those go to control the eyes. Now, those see these white bands of tissue underneath it, right here? Those are the cerebral peduncles, here and here. All right, so you got oculomotor nerve, cerebral peduncle. Now, one other thing I want to show you is this. See this kind of brownish area right here? It's kind of soft to the touch with the probe. Right there, that is the infundibulum. That's where the pituitary gland was attached a few moments ago. So that's the infundibulum. Infundibulum, all right? Now, as we keep coming back, all right, keep coming back, you see it rises up right here? See how there's a risen area right here, right? See that risen area right there? That's the pons, P-O-N-S, the pons, all right? And then as we come back further, right in this area, this is the medulla oblongata. Now, before I get to that medulla and go into that in much detail, let me show you a couple other nerves. If you look down here, now I know these look like bits of ramen noodles, okay? They're not. But right here, you see that nerve right down there between the cerebrum and by the pons? There's one here, the other one got cut off too narrow. This one right here, that is the trochlear nerve. Trochlear nerve right there. And then if I come back on the medulla oblongata, see there's a little nerve right there, right here. That nerve there, that's on the medulla oblongata, that's the abducens nerve. That deals with a little bit of movement of the tongue and taste and so forth, but that's the abducens. So you've got abducens, trochlear, oculomotor, optic nerves have been disassembled, and the olfactory nerve, which is the olfactory bulb and olfactory tract. Okay. Now, running right down the middle, see there's this depression right down the middle right here? That depression right down the middle is the ventral median fissure. Ventral median fissure. It's a little depression. It's not that deep, but it's deep enough to call it a fissure there. Okay? Now, on the medulla oblongata is right here. There are two kind of, you gotta use your imagination a little bit, pyramidal areas right here. One here, and that's called a medullary pyramid. Another medullary pyramid over here. Right and left medullary pyramids. Actually, that's the right, that's the left. Okay? Now, that's kind of the surface anatomy. Now what we're going to do is we're going to get to the internal anatomy. All right? And this is, we're going to make just two cuts today. And this is the first one, and most vital one. So the one of you and your partners who has probably ever had the least amount of coffee or caffeinated beverage today, you should probably have a scalpel. Okay? You're probably less jittery. All right? But whoever's got, who's ever the most calm of hand, determine who that is, have them take the scalpel. Okay? Have the brain right side up. You're going to take the scalpel. Now, as you touch this, you notice it has like the consistency of a stick of margarine. Okay, it's not that tough of tissue. All right. those, I've actually, when I teach nutrition, I look up, I've had students ask, what's the nutritional content of brain tissue? And for the most part, it's fat cholesterol. And so it's a lot like margarine. Now, what you're going to do is this. You're going to take your scalpel, and what I want you to do is I want you to go right down the midline, right down the middle of the brain, and cut all the way down to the paper towel so that when you get done with this, the brain flops into 2 equal halves. So you can have one, your partner can have one if you so choose. Okay, just like that. Ooh, good one. Okay. Do your best to cut right down the middle. Okay, like so. When you get done, then what I want you to do is flip over to page 295 in your lab manual. Flip to page 295. On the bottom of page 295, you'll see a picture that should be very similar to what you're seeing on your dissecting pen. All right? Now, let's take a look and see what we see. Now, okay, here we go. Looks just like this. Okay? And as we go through this, you got the cerebrum. And back here, Arian, this structure right here is the what?
Arbor vitae.
Okay, yeah, the white is the arbor vitae. The whole thing is the cerebellum. But you can really see that arbor vitae, right? See how it looks like a tree that's been snowed on, right?
Ours is squished.
And so that's the arbor vitae right there. That's the myelinated area. Your cerebellum helps you with your coordination, all right? It's interesting if you were to have like a 1 or 2 year old cadaver, okay, which is pretty rare, this is very much gray. And if you ever tried to play catch with a 2 or 3 year old, just like a soft ball, not a softball, but a spongy type ball, it's just a joke, right? Because it's going to bounce off their head, their chest, their face, whatever, because they don't do well, right? They're not myelinated, they don't have good coordination at 2 and 3 years of age, all right? We don't have volleyball leagues for 3-year-olds, okay? It'd be a joke, it'd be frustrating. So you got the arbor vitae right there, okay? And this is part of the cerebellum here. Now, if you come up here, and you can definitely see a color contrast, right? You notice how it's very white here, kind of brownish, tannish here. Where it's white, that's the myelin. Now, you see the swoosh, right? See the swoosh right there? That's the corpus callosum. That was at the bottom of the longitudinal fissure. That's the corpus callosum there. Now, you'll see this downward part coming down like here. That is the fornix, F-O-R-N-I-X, fornix, all right? So corpus callosum, that is a tract that connects the right and left cerebral hemispheres together. The fornix is this side. Now, one of the structures that we have listed is what's known as the septum pellucidum. Now, if you think about a septum is a wall. Some of you maybe have heard of people with a deviated septum, like in their nose, right? It kind of bends one way or the other. A septum is a wall, and right here there's a wall of tissue. Now, it's only about a 32nd of an inch thick. So if I take my probe and just poke right underneath the corpus callosum and then run it back like this, under the corpus callosum, it opens up, just like that. See that? Okay, how it opens. All right? This little thin flap right here, that's the septum pellucidum. It's not very thick. All right? Now, if I open this up and kind of spread it, you see how it's a cavity, right? That cavity back there, that is the lateral ventricle. Okay, that's the right lateral ventricle. Okay?
Flap.
The flap is the septum pellucidum.
I think
That's the flap right there. That's right there, what's left of it. That's
Wouldn't
the
that
lateral
be
ventricle.
Wouldn't that be the left?
This is actually the right, because I'm on the right side of the brain. The left side's the other one. Okay, I'm working on the right half. The septum pellucidum is the flap here. Lateral ventricle's here.
Now I'm really confused.
Now, do you see right here under the fornix? See the fornix right there? There's the fornix. Do you see underneath it right there? You see there's a blood vessel right there. See that? That's the choroid plexus. That makes that cerebrospinal fluid. Now, in here, there's also a choroid plexus. If you look deep in there, see all this kind of wet red stuff right there? See that? that's the choroid plexus there also, all right? Now, if you take, and let me do this, see this center area right here? This is the thalamus right here, the intermediate mass of the thalamus. Do you notice around it, there is a little, and I gotta carve it out because I didn't get right on. There's a little moat, a little depression. See this depression right around it right here? Okay. That depression where I'm running that probe right there, that's the third ventricle, okay? So you got lateral ventricle, third ventricle. The third ventricle leads into the cerebral aqueduct. See how there's a channel right here? That's the cerebral aqueduct right there. Into the fourth ventricle. Okay, so let me go over that again. This cerebrospinal fluid is made every day. It's made up here in this choroid plexus in the lateral ventricle. Now, I didn't show you this a moment ago, so I'm going to do it this time. Take your probe with me, those of you who have the probe. Go underneath the fornix and finagle it just a little bit. There's an opening right here, and you see how my probe came out? The tip of the probe is now in the lateral ventricle. See that? Goes right underneath that fornix. The opening through which it passes is the interventricular foramen, or the foramen of Monroe. Okay? Remember a foramen, like the foramen magnum? A foramen is a rounded opening. So the fluid passes through that, circulates around here, third ventricle, passes through this cerebral aqueduct, which is right here, the aqueduct, down between the cerebellum and the medulla right here, that's the fourth ventricle, right here where the probe's at, and then passes, and you can see there's a little channel right down through the spinal cord, that's the central canal, okay? All right? That's the flowage pathway, how the cerebrospinal fluid flows through the brain and into the spinal cord. Okay? Now, a couple of other things briefly. This is the pons, actually this is not the pons, this is the thalamus. Thalamus right here, sorry about that, thalamus. If that's the thalamus, Brandon, that means right here is what?
Hypothalamus.
The hypothalamus, you got it. Okay, that's the one that probably trips up more people than anything. Thalamus, hypothalamus. Hypo means below. Okay. Thalamus, hypothalamus. Okay. Thalamus, hypothalamus. And then, yes, Brandon.
So the third ventricle would be right in between the, oh, okay.
Right, yeah, it's right around here. So if I were to point here or